Welcome to this tutorial video. So in today's session, as you can see, we're just going to go through some questions in human anatomy. So it's for those that are preparing for the upcoming exam. So we'll just answer some multiple choice questions, then we'll also answer some essay questions. Then in the next video, we'll look at some diagrams that you can come across in case in an exam. So make sure that you subscribe to the channel so that as I upload that next video on the diagrams in the past paper questions, you are well updated. So this is human anatomy, one of the past papers from the University of Zambia. So just go through some questions, okay? Some questions. All right. So you can see, I hope you're able to see. I know that the words may be blurred and everything, but I hope you are able to follow through. I'll be reading the questions and I read the options, then answer them like that. Okay? So don't worry about the numbers, they are jumbled. Don't worry, just follow through the questions. So, question 33 was as follows. Dash are connective tissue strings that attach papillary muscles to cusps of the atrioventricular valves. Dash are connective tissue strings that attach papillary muscles to cusps of the atrioventricular valves. So this question is falling under the cardiovascular system. Specifically, it's looking at the structure of the heart. So the first option is that musculi pectinate, and then B, that is trabecular canine, C, cordia tednae, and then D, that is the sulcus terminal. So before maybe I answer, what do you think is the best option here? So the correct option here is supposed to be the cordia tednae. So the cordia tednae, this is a connective tissue that is attaching the papillary muscles to the atrioventricular valves. So those valves in the heart, like the tricuspid valves, the mitral valves, they are the ones which we collectively refer to as the atrioventricular valves. So they are connected, okay, to the papillary muscles of the heart by what we call the cordia tednae. If you have not seen this, I would recommend that download the picture of showing the internal structure of the heart. You will see that the cordia tednae, this is the connective tissue that is attaching the papillary muscles, okay? The papillary muscles to what? To the atrioventricular valves, cusps of the heart. So that is the correct answer here, which is C. Then question 34 is saying that the heart lies in the A, superior mediastinum, B, anterior mediastinum, C, middle mediastinum, and then D, posterior mediastinum. Now, this is also falling under the cardiovascular system. So basically here, they're just trying to assess if you understand the specific location of the heart. So we looked at the cardiovascular system and then we also talked about these things. So the correct option here is supposed to be in the middle mediastinum. Because if you're talking about the middle mediastinum, this is basically a region that is collectively found in the thoracic cavity. And it's divided into the superior mediastinum, the anterior mediastinum, the posterior mediastinum, and the middle mediastinum. So the word mediastinum, it doesn't necessarily mean middle. No, it's just describing a region which you find in the thoracic cavity. And specifically, the heart is located in what we call the middle mediastinum. Okay, so the correct option is supposed to be C. Then question 35 is saying that concerning the superior vena cava, so it's asking us to identify which option is correct here. So concerning the superior vena cava, A, it is formed by union of the subclavian veins. So we're talking about the superior vena cava. So the superior vena cava, this is a vein, right? It's the vein that is collecting deoxygenated blood from the upper parts of the body. That blood is above the heart. So if you're talking about this particular concept, since you're talking about the superior vena cava, formed by the union of the subclavian veins, this is not correct because the subclavian veins, most likely, they're a bit of the clavicle bone. Okay, so this is not correct. Formed by the union of the subclavian vein and the azygos vein, this is not correct. Formed by the union of the brachiocephalic veins. Remember, I've always emphasized that in human anatomy, each and every topic that you cover, be interested in the diagrams. So in the notes that I had sent for those that are with SPA, okay, for those that are with us with the tuitions, in the notes that I sent, you look for this, you'll find that the superior vena cava is formed by the union of the brachiocephalic veins, which are both from the right side and the left side. And then they mix together, then they form the superior vena cava, which is basically transporting deoxygenated blood from the upper parts of the heart or of the body to the what? To the right atrium, then of course right ventricle, then the circulation is going to continue. So the correct option here is supposed to be C. Then 36, still again under the cardiovascular system, it's saying, which of the following is an elastic artery? Which of the following is an elastic artery? Remember, there are different types of arteries. There are elastic arteries, okay? There are muscular arteries and the likes. So here you must identify, because we looked at the arteries falling under what we call blood vessels topic, okay? And then there are different types that we have here. So let's see. The pulmonary artery, the radial artery, the brachial artery, the femoral artery. So if you assess, because you know the blood vessels are lined with different layers. There's the tunica intima, there's the tunica media, and the tunica externa. Okay, so if you're talking about a blood vessel being elastic, it simply means that that elastic tissue that it has, it gives it the ability to expand a lot or to withstand pressure. And then we said that elastic arteries are close to the heart. So the pulmonary artery here is going to qualify to be called what? An elastic artery. Why? Because it's close to the heart. So all the blood vessels, in short, all the arteries, not blood vessels, but all the arteries which are close to the heart, like the aorta, the pulmonary artery, they have got elastic arteries because of the pressure that is exerted on them because they are close to the heart. So when the heart is pumping blood, they must have elastic tissue that is enabling them to expand. As a result, they'll be able to withstand that particular pressure. So A here is the correct answer on question 36. Then question 37 is saying that concerning blood vessels, so in general, concerning blood vessels, A, all veins carry deoxygenated blood. That is not correct, because the pulmonary vein carries oxygenated blood. All arteries carry oxygenated blood. That is still not correct, because the pulmonary artery carries deoxygenated blood. All veins carry blood towards the heart. Let's keep that. Then D is saying all arteries carry blood towards the heart. So which ones carries blood towards the heart? All veins carry blood towards the heart is correct. So C is correct, because all the veins carry blood towards the heart. Then question 38 is saying dash are capillaries which have a basement membrane which is not intact. So capillaries, remember capillaries have got just two things which are special. They only have one endothelial layer, and then with a basement that is surrounding the endothelial layer, protecting it, of course, covered by pericytes. And then here they are saying that dash capillaries have a basement membrane which is not intact. So if the basement membrane of this capillary is not intact, it simply means that it has got pores, it has got holes inside. Then it simply means that that becomes a discontinuous capillary, because the basement membrane is not intact. It's like there are gaps in between. But if it has the basement membrane was intact, then it only has small pores. We're going to refer to that as what? Fenestrated capillary. Okay, continuous, you find that the basement membrane is intact, it's continuous. Okay, so A here is the correct answer on question 38. Discontinuous capillaries have a basement membrane which is not intact. Okay, then question 64 is saying maturation of spermatozoa occurs in the A, seminiferous tubules, B, the epididymis, C, the red testis, and then D, the vas deferens. So specifically, some of you may think that the seminiferous tubules, they are the ones, it's the correct answer here, but the correct answer is the epididymis. Because in the seminiferous tubules, yes, sperm production starts to take place there initially, but maturation or post-morphological action on the sperms takes place in the epididymis. So the epididymis there is the correct option. Then question 65 is saying, which of the following cells of the testes produce the hormone testosterone? So this is falling under the endocrine system, or you can think it's falling under the male reproductive system. So which of the following cells of the testes produce the hormone testosterone? So the correct option here is the Leydig cells. So the Leydig cells are the ones that produce testosterone. Take note that sometimes you can get confused because the Sertoli cells, they produce what? They produce the actual spermatozoa. Okay? So the ones that produce testosterone, we call them the Leydig cells. Okay? Then question 66 is saying, which of the following hormones is responsible for ovulation? Which of the following hormones is responsible for ovulation? So ovulation, now we're talking about the actual day or phase in which the female ovary is releasing the ovum or the female egg. That is the ovulation. Is it A, saying progesterone, B, luteinizing hormone, C, follicle-stimulating hormone, D, testosterone? Of course, we're going to remove D automatically. Follicle-stimulating hormone. No, this one is just involved in the follicular phase where the follicle is developing. So the correct answer is supposed to be B. Luteinizing hormone is the one that brings about ovulation. We usually say the surge or the increase in luteinizing hormone, specifically in the 28th day cycle at day 14, brings about ovulation. So it's LH, or luteinizing hormone. Question 67 is saying the female reproductive system. So they're just generally assessing your knowledge on the female reproductive system. A, the epithelial cells of the uterus have microvilli. Microvilli, no. Microvilli, you wouldn't find it in this particular region. Okay? The uterine tubules epithelial cells are ciliated. Why would the epithelial cells of the fallopian tube be ciliated? Okay? And then the uterine glands are simple tubular. So the correct option here is supposed to be D, which is saying that during menstruation phase, okay, the parts of the functionalities of the endometrium is lost. Remember, when you are talking about menstruation, you need to keep in consideration what we refer to as the endometrium or the inner lining of the uterus. So we say that inner lining of the uterus has got what we call the stratum functionalis. And during menstruation, it's that part, okay, it's that part of the stratum functionalis which is lost during what? Menstruation. Because the blood vessels, they are directly attached to the stratum functionalis. It's the one which is shed off during menstruation. Okay? Then question 68, it's saying, in which of the following structures does fertilization occur? So remember that, of course, the female reproductive system, or the internal organs specifically, the oviduct, the uterine tubules, the fallopian tube, it is long, of course, but there is a specific region or area where fertilization most likely takes place. We refer to that as what? The ampulla. The ampulla is the correct region where fertilization takes place. Then question 69, because the fimbriae, you're talking about that funnel-like structure, as well as the infundibulum. He's talking about that funnel-like structure, which is more like a funnel, which basically propels the ovum when it's released, bring about assimilating it. Okay? But the ampulla, that's where fertilization usually takes place. So you find that this particular spermatozoa, it's going to move up from the vagina, cervix, up to the uterus, then it's going to locate the ampulla. That's where fertilization is going to take place. Then 69 is saying, which of the following statements is false concerning oogenesis? So, which statement is false concerning oogenesis? So, oogenesis is basically a process that involves the formation of the female ovum or the female oocyte. So, let's identify here which statement is not correct. Ovum is formed in the ovary from oogonia. The ovum is formed in the ovary from oogonia. This is not correct because no oogonia is present at birth. Because if we're talking about the ovum, you're talking about the active mature female oocyte. So there is no oogonia present at birth. So where will this come from? So E is not. So let's see first. Let's first assess this. Which of the following statement is false concerning oogenesis? So the ovum is formed in the ovary from oogonia. Okay, since here it has not specified whether it's during birth or not, it's just saying the ovum is formed in the ovary from oogonia. The fact that the ovary contains oogonia as early as 6 weeks of pregnancy, then E is going to be maintained as correct. So E is just okay. Then F, primary oocytes and its follicular cells constitute the primordial follicle. Yes, that is true. Okay, then primary oocytes begin its meiotic division before birth. That is correct. Before birth, the primary oocytes begin their meiotic division. Then oogonia remains dormant in the ovary until puberty. Oogonia remains dormant in the ovary until puberty. That is now the false one, because oogonia is not present at birth. So at birth you not have oogonia, you have what we call the primary oocytes. So H is the false statement here. So don't confuse the first statement, which is talking about the ovum is formed in the ovary from oogonia. That is correct, because if you're talking about the ovum being formed, it starts from oogonia. Even in as much as it starts before birth, but it starts from oogonia, then that's when the ovum is going to be formed. So H here, which is saying oogonia remains dormant in the ovary until birth, is the false statement. Then 70 is saying concerning the secondary oocyte at ovulation. Concerning the secondary oocyte at ovulation. The secondary oocyte is surrounded by the zona pellucida and corona radiata. Let's see, well, we are supposed to look for a correct statement here. Then F is saying the secondary oocyte is in its meiotic division at prophase 2. So the secondary oocyte is in its second meiotic division at prophase 2. That is not correct. That's not correct. Because if you're talking about second meiotic division, the secondary oocyte is not at prophase 2 when it's undergoing second meiotic division. No. Because the secondary oocyte it transforms or differentiates into what we call the Graafian follicle. Okay? It's the one that goes into the meiosis 2 part. The secondary oocyte has layers of follicular cells called granulosa. The secondary oocyte has layers of follicular cells called as granulosa. The secondary oocyte has completed its second meiotic division. So let's assess this. Let's assess this particular concept. So concerning the secondary oocyte at ovulation. So here we are talking about the secondary oocyte at ovulation, at the actual day of your ovulation. So think about this. Is A correct? Let's see. The secondary oocyte is surrounded by zona pellucida and corona radiata. Yes, this is correct. Because at ovulation, the secondary oocyte is going to be in its meiotic phase, specifically metaphase 2, not prophase 2. So that's why you're finding that F is not correct. Okay? Then the secondary oocyte has got layers of follicular cells called granulosa. No, the granulosa cells, they will be in the follicular phase, while we've got the granulosa cells and the thicker cells. So E is the correct option here. Then talking about question 39, it's saying which of the following is true concerning the conduction system of the heart? Which of the following is true concerning the conduction system of the heart? So Purkinje cells are part of the system. The atria receive the impulse earlier than the ventricles. So, in multiple choice questions, it's all about you being able to identify the perfect answer. There are some answers which may be correct, but you need to identify the perfect answer. So me, just by reading B, the atria receive impulse earlier than the ventricles. That statement is correct, because in the conduction system of the heart, there is an order in which these electrical impulses move. So firstly, they start from the sinoatrial node, which is located in the right atrium. Okay, in the right atrium of the heart. Then that particular now electrical impulse is transferred to the anteroventricular node, from the anteroventricular node to the anteroventricular bundle, then to the Purkinje fibers or subendocardial plexus. So B here is correct. So we will not go through all the MCQ questions because there are a lot. So for the sake of time, let me just also talk about some essay questions so that you also have an idea of how essay questions, come about or how they are phrased. We're just going to answer about 4 of them. They are basically modified essay questions that, of course, are simple and straightforward. Okay, so those are some diagrams that you can see in this particular paper. This is basically the cell membrane. Okay. This is another reproductive system. The cardiovascular system there, you can see the integumentary system there, that's the skin. You can see there. This is under the urinary system. There's the nephron there that is being shown. The digestive system here, as you can see. So let's answer these questions. So section D has got the essay questions part. So each question here, you said it had 10 marks. Then this was 40 marks in total. So question A is saying that outline Genesis. Outline Genesis. So if they say outline Genesis, you're just not supposed to write one sentence, then you end there. It's 10 marks. So you have to outline. So what is first you need to understand what is oogenesis. So oogenesis is basically the process of forming a female ovum or forming the female sex cells. That is what oogenesis is all about. It falls under the topic of gametogenesis. In my video playlist, if you check, you'll find gametogenesis. And then if you watch that video, you'll be able to answer this question. So if you're talking about oogenesis, you're talking about a process of forming the female ovum or the female egg in the ovary specifically. So since this is a modified essay question, it simply means that you need to outline the steps, the major steps or events that takes place in the formation of oogenesis. Remember, it's important for you to give a brief overview of how this oogenesis comes about. So on your overview, you write first, on the overview, you write to say, they are what we call primordial germ cells from the walls of the yolk sac that arrive in the ovaries at week 6 of gestation. And then those primordial germ cells, those primordial germ cells are the ones that differentiate into what we call oogonia. Oogonia has got what? For the 6 number of chromosomes, it's diploids, 2N. So that will be your overview, your starting point. So you've told them to say, before even start talking about the oogonia itself, it's coming from the primordial germ cells from the walls of the yolk sac that will arrive in the ovaries at week 6 of gestation. And then they're going to differentiate, you're going to convert that particular primordial germ cells, okay, into what we call oogonia. Then now, this oogonia has been formed. So you can now write if you want now, you can now go and write, say, oogonia formation. So basically, if you're talking about oogonia formation, you're just talking about the conversion of the primordial germ cells from the walls of the oocytes forming oogonia. Then there will be what we call now primary oocytes formation. So from oogonia, you're going to form the primary oocytes. So you're going to realize that this oogonia is going to differentiate into what we call primary oocytes. It's going to differentiate into what we call primary oocytes. Of course, this is showing the beginning of meiosis. For you to convert the oogonia into primary oocytes, you've initiated the phase of meiosis, specifically meiosis 1. Okay, meiosis 1. And then after form the primary oocytes, you're going to form what we call the secondary oocyte. Actually, in meiosis 1, where you're converting oogonia to primary oocytes, you also form the first polar bodies there. There's a polar body that is formed. Then you go into what we call secondary oocytes formation. So in secondary oocytes, now you're talking about you converting the primary oocytes to secondary oocytes. And then, of course, we are also going to talk about mature ovum formation. So we started from oogonia being formed from the primordial follicles. Okay, then from there, oogonia is converted to primary oocytes. It indicates the beginning of meiosis 1 there. Then primary oocytes are going to form the secondary oocytes. Secondary oocytes now, they are now in meiosis 2. And then from there now, you find that at puberty, there will be formation of the mature ovum, right? At puberty, because each and every menstrual cycle, ovarian cycle, there should be ovulation. So there will be formation of the mature ovum. Then, of course, you can talk about the polar bodies that are formed in general. Remember, when I was talking about the menstrual and ovarian cycle, I said only one ovum reaches full maturity in each and every ovarian cycle. So you're going to produce other cells. They differentiate into what we call polar bodies, which just basically degenerates. So this is how you're supposed to outline it. Talk about oogonia formation, talk about primary oocytes formation, talk about secondary oocytes formation, then the mature ovum formation. That's how you're going to line the formation of the female ovum. Of course, now maybe you can also talk about the fact that there is no oogonia which is present at birth, because at month 5 of fetal life, you would have formed all the primary oocytes. Then that primary oocyte is going to be arrested at prophase 1 of meiosis 1. Then during puberty now, that's where now it's going to resume this meiosis, where now there will be formation of the secondary oocytes. Then this secondary oocyte now will be arrested at metaphase 2 of meiosis 2 until fertilization takes place. If there's no fertilization, it's just going to die or generate in secondary phase of meiosis 2 in metaphase 2. So those points must come out. Then question 2 is saying compare and contrast. Compare and contrast the what? The skeletal and the cardiac muscle cells. So here you have to compare and contrast skeletal and cardiac muscle cells. So this is about 10 marks. This is about 10 marks. So maybe you're supposed to talk about 10 differences that you can talk about in terms of the skeletal muscle and the cardiac muscles. Okay? So think about it. What can you say here? What can you say if you were asked to outline the differences between the skeletal muscles and the cardiac muscles? So since they're talking about you talking about you're comparing and contrasting. So maybe when comparing, you talk about the similarities first. You talk about the similarities. So you find that both skeletal and cardiac muscle cells, both of them, they are striated. Both have got the sarcomere and the microfilament, which brings about the striation characteristics. Then both of them have got contractile function, meaning both of them they contract. So both the skeletal muscle and the cardiac muscle, they both what? Contract. So at least before we even talk about maybe the differences, of course I know that a lot of answers are on the differences, but talk about the similarities. Okay? They're striated, they've got myofilaments that are found in their structure, then they also what? Contract. Then talking about location. Now you start now talking about the contrasting part, the differences. Location. Skeletal muscles are located or they are attached to bones. Okay, these they are attached to bones. Okay, then the heart muscles or the cardiac muscles, they are found in the walls of the heart, specifically what we call the myocardium layer of the heart. Then talking about control, do skeletal muscles move on their own or not? So skeletal muscles are under what we call voluntary control. Then cardiac muscles are involuntary. Don't stop your heart from beating. It will keep on contracting and relaxing. So skeletal muscles are under what we call voluntary control. Cardiac muscles are under what we call involuntary control. Okay? So talking about striation, we said both different types of muscles, they are striated. Okay? Talking about the nuclear. Okay, the nuclear. So skeletal muscles have got what we call multi-nucleated nuclei per cell. Then the cardiac muscles have got a single central nucleus. Okay? Then talking about the shape. If you focus on the skeletal muscles, you find that they've got a cylindrical kind of shape, more like a rod, more like a rope-like. We call it cylindrical fiber shape. But if you talk about the cardiac muscles, they are highly branched. They are highly branched. Okay? And then if you talk about in the skeletal muscles, you don't have what we call intercalated discs, because the intercalated discs are the one that brings about what we call a cardiac syncytium, where muscles of the heart they function or contract at once. Okay, so you find that the heart muscles or the cardiac muscles have got what we call intercalated discs. The skeletal muscles, they don't have intercalated discs. Okay, so that's one of also the difference that you can talk about. Okay, so then you can also talk about the activity. What's the function behind these two different muscles? So the skeletal muscles, they're involved in movement and posture, while the cardiac muscles, they basically pump blood. From this general guidance that I've given, you can come up with a lot of points that you can use to answer question 2. Then question 3. So question 3 is talking about the classification of glandular epithelia. So remember, this is falling under what we call epithelial tissue. So epithelial tissue is basically a type of tissue that covers both surfaces. It makes glands and everything. And then it's majorly classified into two different components. There's what we call covering epithelia and the granular epithelia. So in the granular epithelia, you're talking about epithelia that forms glands. Now, we are talking about classification of granular epithelia, which can be done on different or using different methods. So, for example, you can classify epithelia based on the number of cells present. If it's in unicellular gland, meaning there's only one cell that is present there. For example, single secretory cells like the goblet cells in the intestines. Okay, those are unicellular, kinds of granular epithelia. Then there are what we call multicellular glands, which have got a lot of cells for coming together, right? Since we're talking about epithelial tissue, okay? So these you would find them, for example, in the salivary glands and everything, and the pancreas. They have got they are multicellular cells. They have got a lot of cells, but the fact that they are glands, we're going to cell them, they are multicellular glands. Then you can also classify epithelia based on the type of secretion. Is it an exocrine gland? Is it an endocrine gland? So exocrine gland, they secrete glands into what we call ducts. Then the endocrine glands, they secrete hormones directly into what? The blood stream. So we've got endocrine glands and the exocrine glands. So that's also a type of secretion that you can of granular epithelia that you can use to classify granular epithelia. Endocrine epithelia as well as exocrine epithelia or glands. Then you also have what we call mixed glands, because there are glands which can be both endocrine and the exocrine. Like for example, the pancreas. Okay, the pancreas is both an endocrine and exocrine gland. So you can classify it in terms of endocrine gland, exocrine gland, and the mixed gland. Apart from that, you can also classify epithelia based on the type of secretion that is performing. Is it merocrine secretion? Is it apocrine secretion? Is it holocrine secretion? For example, if we're talking about merocrine secretion, we're talking about secretion of content from these epithelial or granular epithelia via what we call exocytosis. For example, the salivary glands, they just secrete saliva through what we call exocytosis. Then if you're talking about apocrine secretion, you're talking about where some part of the cell is going to be pinched off. Like for example, what happens in the mammary glands when somebody is breastfeeding and everything, some parts of the cell, they are pinched off from the entire cell, then they bring about secretion. Not the entire tissue, but part of it is just pinched off or removed and bring about excretion. Then there's also what we call holocrine secretion, whereby the entire tissue is going to disintegrate or damage or breaking down, not as a pathology, no, but under normal circumstances, and then bring about secretion. For example, the sebaceous glands, you have got what we call holocrine secretion. Then there's also a question 4 which is talking about comparing and contrasting meiosis and mitosis. This is a very simple one. So if you're talking about mitosis and meiosis, of course, you know that meiosis only takes place where in sex producing it takes place in the male and female gametes only, in the sex cells. And then mitosis, it takes place in what we call somatic cells. Of course, if you go in details, there's a part where the sex cells, they also perform mitosis. For example, if you're talking about gametogenesis, there's that particular phase, but mainly meiosis is only involved in sex cells, while mitosis is involved in what we call somatic cells. Any cell that is not a sexy cell. So if you're talking about the differences, we talk about one, the purpose. What's the purpose of meiosis? What's the purpose of mitosis? In mitosis, the purpose is bringing about growth, development, and asexual reproduction. Okay? We're talking about growth and development because the cells must grow, they must divide. But we're talking about meiosis, it's talking about sexual production of gametes. So production of gametes. Okay? Then if you're talking about mitosis, so if you're talking about the mitosis only has one cell division cycle or phase. But in meiosis, there are two phases that take place. There's meiosis 1 and meiosis 2. And then talking about the number of daughter cells that are produced. Mitosis produces 2 identical daughter cells, while meiosis produces 4 genetically different daughter cells. Talking about the number of chromosomes. In mitosis, you produce diploid number of chromosomes, while in meiosis, you're going to produce haploid number of chromosomes, or 23. Okay? So, talking about also genetic variation. The resulting daughter cells that are being formed, are they going to be different from the parents? In mitosis, there is no genetic variation, but in meiosis, there is genetic variation. So, meaning the parent cells are not the same as the daughter cells. Okay? Then is crossing over present in mitosis? The answer is no, because crossing over is the one that brings about genetic variation. But crossing over is present in what? In meiosis. Okay, specifically in prophase. Okay, so that is basically some of the differences that you can talk about in terms of meiosis and mitosis. So from the few questions that we've covered, I hope you'll be able to subscribe to this YouTube channel so that as I upload another video on these particular questions, you're able to follow through.